Well, hello, everyone. For those of you that don't know me, I'm Kate Laskowski, and it's my great pleasure to introduce Sanaa Khan today. And so Sanaa is a PhD student in my lab. She's in her third year, and so she's going to present to you on some data that she's already collected and then stuff that she wants to do. So a little bit of her background. She did her undergrad and master's at the National Institute for Science Education and Research in India. And the thing that most impressed me about Sanaa when you interviewed, she interviewed in 2022, and so it was all completely remote. And she was still based in India at the time. And so I remember having a conversation with her, and it was like 3:30 a.m. And so the fact that she could hold a cogent conversation about like theory and how she would test it and all these things really, really impressed me. And so we brought her over, obviously, and she's been a great addition to the lab. She's going to tell you today about the feedback between males and females and making mating decisions. And the other thing you're going to see in this talk that keep an eye out is for Sanaa is one of our resident creative artists in our lab. She's an incredible artist, drawing lots of things, writing lots of things, reading lots of cool things. And so we'll get to see all the many facets of her creative mind in this talk. So take it away.
I am deeply nervous to be doing this talk, but also so excited to be talking about my research. And my research is about exploring feedbacks between female mating strategies and male charts. So when you're studying sexual selection,
I have to click on the screen.
yeah.
Yeah, there you go.
Okay, when you're studying sexual selection, you're thinking about interactions between males, you're thinking about interactions between females, and between males and females, obviously. So male competition, female choice, male choice, and maybe female competition as well. We know a lot about male competition. Male competition research, as old as sexual selection itself, one of the first things Darwin came up with. And it produces some pretty ostentatious results, like these elephant seals fighting to the death sometimes. And we also know a fair bit about female choice. An example here is these bowerbirds. The males, of course, put up these wonderful displays, and the females then choose among the bowers of these males. So a lot of information on female choice as well. Now, male choice, slightly less information, but we still have studies of male choice. Example, a lot of pipefish and seahorses have male choice. Female competition, again, less information, especially when thinking about direct competition for access to mating opportunities. But again, we have examples of that. For example, cassowaries, which are a complete sex role reverse system where the females occupy these large territories that they defend against each other, and they fight for access to males. And in many ways, mating systems are a spectrum where we have male competition and female choice at one extreme, and female competition and male choice on the other extreme. There's a reason it's called a sex role reversal system. But these things exist together. You can have male competition, female choice, male choice, all together or some combination of these things. And often, because signals for male competition and female choice are so strong, because they're ostentatious, but also because we have our own biases built upon decades of research. The other signals, female competition and male choice in these systems, become more obscured. But there are indeed examples. There are antelope, where these are two females fighting against each other for access to the best male, even though this is a lekking system. So males are displaying on leks, and usually females are just going around and choosing the males. But here, females are also competing for access to certain males. And this is an example of, the other picture is an example of a snipe where males will, which is again a lekking system, where males will exercise some male choice. Which brings me to my question. In non-sexual reversed systems, I'm interested in my first chapter to look at female competition. For my second chapter in the same system, I wanted to explore male choice and what are the things that might be affecting them. And then, of course, I want to look at the interaction between these two things for my third chapter. What should I do this study in? What system could I possibly use? What if I had the ideal system? A system that has either strong male competition or a female choice. Maybe I can really tell what the quality difference between the females is from the male's perspective. And maybe females have a really big incentive to compete. Guess what? I do have this ideal system. And she's the Amazon Molly system. So that's the Amazon Molly. And she shares her range with Atlantic Mollies and Sylvan Mollies. So this combination of the female Amazon Molly, and they're all female, and then the male Atlantic Molly and Sylvan Molly, how they choose. This is the system I'm interested in exploring and asking my questions. So, who is she? A little background on the Amazon Molly. So the Amazon Molly is a hybrid. She is the result of a single hybridization event between a female Atlantic Molly and a male Sylvan Molly, which led to her. This happened only once. We cannot generate Amazon Mollies anymore. And she is a sperm parasite. So Amazons reproduce by a process called gynogenesis. So in this example, I'm using an illustration of an Atlantic male. The male contributes sperm to the Amazon. But through a somewhat not well-known mechanism, the Amazon excludes the genetic material from that sperm and produces only clones of herself. So in this system, the Amazon has incentive to mate with males to get access to sperm, but for the males, any sperm that he invests in the Amazon is wasted. There are no direct genetic benefits for the male from mating with an Amazon Molly. So this was my question. These were my 3 chapters. And here I'm going to explore these in the Amazon Molly. Female competition and mating strategies in the Amazon Molly. Male choice, how should Sylvan and Atlantic Mollies choose, and the interactions between them. Which brings me to my chapter 1. What's a fish to do? So she has this situation where she needs to get sperm, but really it's maybe not in the male's best interest to mate with her. And I think maybe she can do a number of things. Maybe she can use a behavioral strategy. She can try to be aggressive against females of his own species. And maybe that will drive away females. Maybe that'll give her direct access to the males. Or maybe she uses some kind of physiological strategy. So from the literature, it appears that males have a preference for larger females. So maybe if the Amazon grows bigger faster, she's able to take advantage of that sensory bias and acquire more mating opportunities with the male than she would otherwise have. To answer the behavioral strategy question, before I can start talking about aggression, I have to ask another question, which is, can Amazons even discriminate between clonal sisters and heterospecific females? Do they know the difference between their own clones and their parental species? So to do that, I did this kin preference assay, where I had 1 focal fish, and my sample size for this experiment was 50 fish. And she was given a choice between 2 fish, one that I call the stimulus or treatment fish, and the other one is the control fish. So this is the Amazon in the middle. The control fish is usually a sister clone. A little bit of a clarification on sister clones and non-sister clones. Amazons are a clonal lineage are a clonal species that was generated through a single hybridization event. But over time, they've of course accumulated some genetic mutations, and there are several clonal lineages. So the control fish is always a sister clone, as closely related to the focal Amazon as is possible. The stimulus fish is sometimes another sister clone in a control condition. Could be a non-sister clone that's still a clone, but maybe slightly less related. Or it could be a Sylvan Molly which is another species, another species entirely. So my predictions were hypothesis 1, Amazons prefer to hang out with closely related kin. And this was supported by previous studies, and it was something that I was trying to replicate. So what I expect is on the y-axis, we have time spent with the stimulus fish. And on the x-axis, we have the various treatments. So I expect that she wants to hang out the most with her sister clone and least with the sailfin, who she is related to, she's not that related to. On the other hand, it might be possible that Amazons prefer to hang out with less related fish in order to maximize mating opportunity. So in this case, I would expect the exact opposite result, where she spends the least amount of time with a sister clone and the most amount of time with a sailfin molly. What were my results? My results supported hypothesis 1. Amazons prefer other Amazons over sailfins. So they spend the most amount of time with their sister clones, and they spend significantly less time with sailfins. So this replicates my hypothesis 1. So now that I know that they can indeed tell the difference between sister clones and heterospecifics, the next question is, are they using aggression as a potential mating strategy? So is female aggression a mate acquisition strategy in Amazon mollies, or are the clones attacking? And for my question, I'm interested in the mating context. So in addition to this is, of course, aggression between Amazon and Atlantic mollies, but also does the context that these mollies exist in matter? So does the presence of a male or a female of another Amazon female matter in terms of the aggression these fish have towards each other? So this was my experimental design. So I had a tank where an Amazon molly was paired up with an Atlantic molly. They were both size matched. And then I had a soda bottle that had a different fish that was acting as like a social treatment or a spectator. So the mating context, mating context is when there is the Atlantic molly male. And I would expect that maybe they're competing for the access to this male. They're able to see and smell the other fish through this soda bottle. And the non-mating context is when the other fish, the social treatment fish, is an Amazon molly. And my sample size for these experiments per treatment was 20 fish. So my expectation is that the amount of aggression depends on the mating context. Amazons are the ones that are being more aggressive, and they are less aggressive in a non-mating context when the spectator fish is an Amazon, and they are more aggressive towards the Atlantics in a mating context when the spectator fish is an Atlantic male. What was the reality? I had one data point where the Amazon was being aggressive towards the Atlantic, and all other situations, she was zero aggression. She was just being beat on by the other fish. So the clones are not attacking. Only 1 out of 40 Amazons was actually the aggressor. But what about the Atlantic mollies? Did I learn anything about the aggression of the Atlantic mollies through this experiment? And maybe I learned something. So Atlantic mollies might be affected by Amazon spectators. So this on the y-axis now is aggression by the Atlantic. That's number of bites, number of chases that she performed. And on the x-axis, we have the non-mating context where the spectator is an Amazon, and a mating context where the spectator is an Atlantic male, so a male of the same species. So the Atlantic molly did appear to be more aggressive in the presence of an Amazon, and slightly less aggressive in the presence of a male of her own species. These results are actually not significant, so I might need a larger sample set to really tease apart what the relationship between these things is here. So maybe if aggression isn't the best strategy for the Amazon, maybe she has to use the physiological strategy. Maybe the way to do it is to grow big fast. And this is just, I don't expect these curves to look exactly like this, but this is sort of a demonstration of what I think would be happening, where the Amazon molly is just ramping up her growth, trying to like get big first, as compared to the sailfin molly. So this is a summary of my results. So Amazons can distinguish between sister clones and heterospecifics. Amazons are not more aggressive than Atlantic mollies. So maybe Amazons are unlikely to use aggression as a mating strategy against Atlantic mollies. Lastly, aggression in Atlantic females may be affected by the presence and perhaps the density of Amazons found in her environment. I'm not quite finished with my aggression experiments. I would like to go back and maybe see how this plays out in the other species, the sailfin molly. But for now, I've moved on to my chapter 2 experiments. So I did chapter 1, female competition and mating strategies. And for chapter 2, I am going to explore male choice. So how should a sailfin molly choose when he has this choice between an Amazon molly, no direct genetic benefits, and a sailfin molly who would give him the expected genetic benefit from mating with a female of his own species? And why do the males even mate with Amazons? The fact that Amazons exist to this day means that the males do mate with them. Mating is expensive. There's opportunity cost when a male mates with an Amazon female. There's sexually transmitted diseases. There's energetic costs. So why not just stick to your own species? And maybe the answer lies with signal detection theory. Signal detection theory basically says that when two signals are very closely related, so here, whatever cue that the male is using to recognize Amazon mollies and whatever cue he uses to recognize a female of his own species, there will always be some region of overlap between them. And the male will have his threshold that he uses to decide which one is the sailfin molly and which one is the Amazon molly. Here, the threshold is indicated by the dashed blue line. And that leads to certain errors. So if he sees an Amazon, decides to leave, that would be correct rejection. And that's good. You saw an Amazon, did not mate with her. You didn't waste your sperm energy doing this. If he sees a sailfin molly though, and he decides to leave because he mistakes her for an Amazon, that's an error, that's misdetection. If he sees an Amazon and decides to try and court her, that's false alarm. That's indicated by these little red stars, this region of overlap. And if he sees a sailfin molly and decides to court her, that is correct detection. The issue is because there's this region of overlap, false alarm and correct detection are correlated with each other. So you can't really increase your probability of finding a sailfin molly and mating with her correctly without increasing your probability of false alarms, which is mating with Amazon mollies. So maybe that's why Amazon mollies continue to get mating opportunities with males. But does the male always choose the same way, or are there things in his environment that could affect male choice? And maybe there are. Maybe the developmental experience that the male has, what kind of fish that he grew up with, I guess, can affect his ability to tell apart these two females. Maybe his latest mating experience also biases him towards one or the other kind of female. And maybe the state of sperm limitation, how many gametes the male currently has to invest in his next mating opportunity, have some effect on his choice between Amazons and sailfin mollies. So speaking of the role of development and mating experience, so, and calling back to signal detection theory, so these probability density functions, this overlap, it's possible that the development and mating experience actually determine what these distributions look like for the male. So maybe if you had more information from the get-go, these distributions wouldn't have such a large region of overlap. And what I expect to happen is that, so if you're thinking about association time with Amazons, and we have these three different treatments where some males were raised in an Amazon biased environment, some males were raised with no bias, equal proportions of Amazons and sailfins, and some males were raised in a sailfin biased environment. What I expect is maybe which female they encountered most during their developmental experience biases them towards that female. So males, so the red dot, males that grew up in an Amazon biased environment, they have a preference for Amazon. If I did a binary choice test, they would spend most of their time hanging out with the Amazon females, versus males that were raised in a sailfin biased system might be able to choose more correctly and hang out with sailfins most of the time. And on the x-axis I have, so this is what I expect when they've had no mating experience. This is the first time they're encountering these females. So when it comes time to have a second mating experience, they've already gained some information from mating with these females. I expect them to maybe get better at choosing the sailfin. So the ones that were raised in an Amazon biased environment, the ones that were raised with no bias, both of them gain new information from this mating experience, and they adjust their choice accordingly. So if mating provides them with new information, this is what I expect to happen. Next, thinking about the role of sperm limitation. So being limited and how many gametes they have to invest in their next mating experience, what kind of effect does that have? Again, when we think about it in terms of signal detection theory, I imagine what might be happening is that the threshold itself, the threshold is changing. Their distributions are maybe the same, but the risk they encounter when mating with the risk of mating with an Amazon and then not having those gametes for the next mating opportunity might be too much for them, and they might change their threshold. So here in this diagram, they're actually moving towards the left. And maybe when they're sperm depleted, they're like, let's just find someone to mate with. I have like one opportunity to mate. Maybe I don't survive to the next time step. I won't be so choosy. But it could also move in the other direction where my gametes are already limited. If I invest if I mate with the Amazon, it'll take me a really long time to recover my gametes, and I might die during that time and not get any reproductive benefit in the future. So I think it can go either way. So males might respond by becoming less choosy. They have limited gametes, try to mate with any female they can, because because false alarm and correct detection are correlated. So they are increasing their chances of mating with the sailfin, even though they're also increasing the chance of mating with an Amazon. However, if it's really expensive to recover those gametes and an incorrect mating could set them back if they have a really long refractory period, maybe they want to be more prudent with their choice and be less choosy when they are slightly sperm depleted. Become more choosy when they're slightly sperm depleted. So this is what I expect for when just thinking about development experience and mating experience, and then thinking about state of sperm limitation. But I would also like to know about the interaction between development experience and sperm limitation. Bear with me, this is kind of a complicated looking diagram. And I hope I get the fish numbers necessary to be able to do this. So I would like to subject my males to 3 developmental treatments: Amazon bias, no bias, and then Silfen bias. I will subject half of my males to like sperm depletion, and half of my males will not be sperm depleted. And these, at this point, the males have had no real mating experience. As soon as their developmental, so the fry will be raised in tanks with different densities of Silfen and Amazon females. Once they are, right before they're sexually mature, their anal fin elongates, but they're still not capable of mating yet. So I've observed a lot of males right at this point while trying to perfect my techniques, and I can recognize when they are juveniles that are about to become male. So I will separate them at that time and house them individually, but in proximity with other males. After that, depletion, no depletion, they'll be given their first binary choice test and then allowed to interact with the females and hopefully mate with the female. Then I will repeat this. They'll get that mating experience, and then I will repeat this and give them their second binary choice test. And I will look at how their choices change between the first binary choice test and the next binary choice test. Look at the interactions between development, experience, and then sperm limitation. So I say a lot of things about sperm depleted, not depleted. How do I control that? I can do this artificially by stripping the males of sperm. In this picture, that's what I'm doing. The males are knocked out with MS-222, and then the sperm is literally squeezed out of them. They have minutes. They are completely depleted. I unfortunately don't have, I did do a little pilot experiment looking at correlations between sperm number and male size, etc., but still in the process of analyzing that data. But I'm just going to talk about some insights that I've gained just by like working with these fish. So first of all, and most importantly, sperm stripping is not lethal to the fish. No mortality among my experimental males, and I had a sample size of 12 males when I was learning how to do this. And from just by isolating them and then doing this process again and again for the course of 4 weeks, I've learned that they can recover sperm, but it takes them up to 1 week, but sometimes it could take them less. But definitely they lose several days to with no sperm, 0 sperm, if they are completely depleted. And lastly, this is no figures because this is just anecdotal information from working with the fish. Males are often sperm depleted when they are housed with females. But often when I go to sample these males, they just don't produce any sperm. And this process of stripping them, if they have sperm, it's going to be squeezed out of them. Don't know any other way to put it. Yeah. So in summary, I'm interested in like, what does sperm limitation mean for Silfen and Atlantic males, and how does developmental mating experience and sperm limitation affect male mate choice? So calling back to purpose of my research, Chapter 1, thinking about female competition and mating strategies. Chapter 2, I'm going to be thinking about male choice. And then for Chapter 3, I want to explore the interactions between female mating strategies and male choice. And to do that, I'm hopefully going to do a little bit of a mesocosm experiment. I know it's technically not a mesocosm if there's no predation, but I'm calling it a mesocosm because I'm putting a bunch of fish in a big tank and I'm seeing what they're going to do. And because I'm doing this mesocosm, I'll also be able to explore the role of the immediate social environment in determining male and female mating behaviors. So this is what I imagine my mesocosm will look like. I'll have some refuges to make the fish comfortable. And then I'll have some females of both species, some Amazon females, some sailfin females, and a sailfin male. These are hopefully what the numbers will look like, but I'm very far from being able to do this experiment right now, so we will see how things change. So hopefully my treatments will look something like this. There's Amazon bias social condition. There's no bias in the proportion of Amazons and sailfins, and sometimes there might be a sailfin bias social situation. And the thing that I hope I'll be able to measure by this time is to look at fertilization success of the Amazon and sailfin males. And this can be done by flushing the female's gonopore with like a saline solution that sort of forces the sperm out of it. Currently in the process of developing this technique, step one for this is to successfully artificially inseminate a fish. And then the next step is to go one step back and like take the sperm out before fertilization happens. It's not succeeded yet in artificially inseminating a fish, but working on it. But this would give me a direct estimate of at least fertilization success and then hopefully reproductive success and differences in reproductive success between the sailfin females and the Amazon females. Some of the other information I can get about the behavior is male courtship behavior between the Amazon and sailfin females, Amazon's aggressive behavior, chasing, tail slapping, or the lack thereof. Then maybe Amazons do courtship interruption. This has again been anecdotally noted in these fish. There, if a male is actively trying to court a female of his own species, the Amazon will sort of interrupt that interaction and swoop in to get that mating opportunity. And then just time spent in the proximity of the males for the Amazons and where the males try to spend their time. And to get all of this behavioral information, I will hopefully be able to use tracking softwares. Our lab has the skills to do this, and I'm hopeful that we'll be able to develop an assay that can track mating interactions between these fish. And we already have the tools available to just track information about where they spend their time. Okay, so this sort of brings me to my summary slide. There, I'm interested in looking at female mating strategies, chapter 1. Then I'm interested in male choice and how the individual state, which is affected by development, mating experience, energetic and gametic limitations, is affecting this male choice. Oops. That's my chapter 2. And for chapter 3, I'm interested in the interaction between these two and also the role of the immediate social environment. Maybe I can look at sex ratios, group composition, kinship. Those are big things, and that's the pipe dream of being able to look at everything in this kind of mesocosm situation where the fish are allowed to interact freely with each other. And lastly, thank you everybody for attending my talks, especially the Skowski Lab and everyone in Pop Bio, especially my Pop Bio cohort. Special thanks to undergrads that have worked with me, Kenneth Damper, Ethan Negus, and Kersley, who was not an undergrad, but a high school student who worked with me in the summer for the YSP project. And thank you everybody for being here.
Super awesome talk. I really thought it was interesting. I was intrigued by kind of how you framed the entire thing, especially I think you'll get at this in the chapter 3. But you were always talking about the male mating with the Amazon molly as a cost to the male. But I was curious, is there any literature of mate choice copying or anything like that?
So there is some literature of mate choice copying there. So it has been found that the sailfin females will mate with the male that's mated with an Amazon, but they would still do the mate choice copying if he mated with a sailfin molly. So it's still, as compared to mating with the sailfin molly, it's still a cost to the male.
Thank you.
I was wondering if it was practical reasons that you chose either an Atlantic or sailfin for different things, or if there's a
Yes.
biological reason to choose one of the other species.
No, it was for practical reasons. I started this research planning to only use sailfin mollies and stick to one species. But several times my experiments just didn't work with that fish. So I switched to the other species. Just for, again, from anecdotally like observing the fish, there is like a huge difference in the way that they behave. Sailfins just tend to be more fearful, more likely to react badly to like a change in environment. So when you move them from the residential tanks to their experimental tanks, they often will just lie on the bottom of the tank and do nothing. But Atlantics, on the other hand, seem to be like a more gregarious species and recover quickly from these changes.
Really beautiful talk. Really good job. Thank you.
Yes. Sorry.
I'm curious, in your last experiment, I might have missed it, but one of the things that was really interesting that you talked about at the end of your first chapter was this idea of body size being really important
Yeah.
for the fish, and you kind of talked about that a couple of times. Do you think, or how are you thinking about that in the mesocosm experiment? I think I just missed it.
So I think to begin with, I would probably have them all be size matched. So body size isn't this other confounding factor, just because there's a lot of interactions happening among all of the fish. So starting with the simplest case is better. So being larger has a lot of benefits, of course, access to resources, potentially access to meals. The body size differences might also better frame the results from my aggression experiments where the Atlantics were more aggressive. Because if the Amazons in general are used to being the bigger fish, maybe why fight when you're size matched? Fight when you're the big fish. So maybe I'm still waiting for my fish to have babies. Yes. Oh, Jeff.
Yeah, thanks. It's really fun to think about. I had a question about what are your predictions for the male choice experiment? Specifically the mating experience. So I guess I agree, it sort of makes sense that the male should get, this slide.
Yeah.
But I think I just didn't understand like why, like what information a male would be getting if it
Yeah.
mates with the wrong fish. Like, is it somehow figuring out, oh, none
Yeah.
of those offspring
So-
are mine?
So the way mating works in these fish is that the males are, it's sort of a coercive mating system. So the males are chasing the females and then trying to mate with her. And one of the behaviors they do before they mate is they nip at the bottom of the on the female's belly. And it seems to be giving them some information about the receptivity of the female. So that's what led me to think that maybe that would also give them important information about, are they getting like the right pheromones from this fish? Is she like the right species? Is she in the right reproductive state? So that's why I think that by actually pursuing a female, attempting to mate, they will gain information, even if they don't succeed in mating. Does that answer the question? Yes.
I think it's just totally fascinating that in this system that the sailfin molly, that the males from this other species will continue to mate with them. I just think that just right there, I think is just sort of wild. And so there's something about that. The male is still attracted enough to this thing, this hybrid.
Yeah.
And so I guess one question I have about this is how far out do you have to go phylogenetically before you find a male that won't try to mate with the Amazon molly? You
So
just-
I'm not certain about that, and I don't know if any papers have looked at that. There have been some studies that look at the other side, whether the Amazon will mate with males of like different peacelet
Right.
species. And she doesn't seem to mate as much if you go outside of her parental species, but she will eventually mate with them. So black mollies, even guppies, will generate like successful gynogenesis.
They will, guppies.
Yeah,
Okay, wow.
yes.
So it's not a very specific thing to the parental species.
Yeah, no, but they seem to have some sort of preference.
Yeah, I was curious. I was interested in your development part where you were saying that maybe depending on group composition, they may have preferences. I was curious, has there been work done, or maybe do you just have like some speculation of in your bias groups? It seems like you had like 4 and 2 for a bias, right? Like 4
Yeah,
Sailfin, 2
Yeah,
Amazon.
yeah.
I was curious, do you have any ideas of if they were all like Sailfin through development and then you switched it to a tank with only Amazon mollies, would they know that that's like an option to mate with, or?
So, okay, so is the question that if you raise them only with Sailfins and then you put them in with Amazons, would they try to mate or not?
Right, like would they know that that's
Yeah,
even
they
an
will,
option?
they will try to mate with the females. Because right now what we have is when we are housing these fish, they are, and when they're fried, they're in their own parental species tanks. And then once they become sexually mature, we move them in with Amazons to fertilize those Amazons. So yeah, that generates successful offspring, but I mean, we're not giving them any other choice. So,
So following on Peter's question, I was wondering a little bit when you were sort of switching in and out the Atlantic and Sailfin mollies, it's Atlantic, right? And Sailfin mollies. Will the Atlantic males mate with the Amazon mollies as easily as the Sailfin males? Because I think usually you have
I
Sailfin males
don't
there.
know. So actually all of the literature so far seems to be on Sailfin molly and Amazon interaction. There isn't really a lot on Atlantic molly and Amazon interaction. So that's what I'm trying to do. Currently in the process of doing a pilot where I'm like subjecting both Sailfin males and Atlantic males to this binary choice test. And I'm also looking at the effect of like having like as much sperm as you're going to generate and then being completely depleted of sperm. Because again, through observing the fish, I think that the males don't try to mate at all when they're completely depleted. They're not, I anticipate they're just not going to choose. So I'm just trying to verify that, and it'll also generate information on species differences.
Because I think that affects the predictions for what the females
Yeah.
would do.
Yeah. So yeah, I don't know if I'll end up getting data on both species. I'm in this phase where sailfish are giving me trouble, so I'm testing out the other species. There's no more questions, I guess I'm done.